Dear students, today I am going to tell you about Sanger's reagent, structure of which is given here. This is 2,4-dinitrofluorobenzene. I am going to tell you about the applications of this reagent, which are determination of N-terminal amino acid of a polypeptide chain and alteration of enzyme activities. So first application is determination of N-terminal amino acid in a polypeptide chain. So it is a solid yellow color crystal. We get it in the form of crystals, and it is prepared by the reaction of 1-chloro-2,4-dinitrobenzene with potassium fluoride in nitrobenzene. In mildly alkaline solution, a reagent reacts with alpha amino acids to produce yellow colored derivatives, which is DNB, that is dinitrophenyl amino acid. These dinitrophenyl amino acids are stable under acid hydrolysis conditions that break peptide bonds. This reaction is valuable in elucidating the structure of protein, which has been successfully utilized by Sanger in England in determining the sequence of amino acids in insulin, and for that he got a Nobel Prize also. Here you can see this amino acid, and this is the NH2 group of this amino acid, and this is reacting with Sanger's reagent. HF is being released, and this is linked to this stable compound DNB amino acid. This is yellow in color, and this can be monitored through a colorimeter or a spectrophotometer. This here the same reaction is shown along with the polypeptide chain, and this is N-terminal end of the polypeptide chain, and this gets bound here, and this portion is released by either acid hydrolysis or enzymic hydrolysis of peptide bonds, and then they can be separated chromatographically and monitored through a spectrophotometer or colorimeter. Other amino acids are colorless, and this derivative is yellow colored. That is why that derivative shows the presence of N-terminal end of any polypeptide chain. Here in this slide I am showing a protein made up of two polypeptide chains which have disulfide bonds shown in black bars. Then these are reduced in presence of dithiotretol or beta-mercaptoethanol, and as a result of which polypeptide chain is shown like this, free from any disulfide linkage. Then mechanism of action is shown here in this slide, where reagent undergoes nucleophilic aromatic substitution with N-terminal amino group of a peptide or protein. ये आप देख रहे हैं यहाँ पे N-terminal end है, ट्राईपेप्टाइड है, R1, R2 एंड R3 तीन अमीनो एसिड से मिलके ये पॉलीपेप्टाइड ट्राईपेप्टाइड बना हुआ है. इसका N-terminal by convention we write N-terminal at the left end and C-terminal end at the right end. So यहाँ पे N-terminal end है और ये free है. यहाँ से न्यूक्लियोफिलिक एरोमेटिक सब्स्टिट्यूशन रिएक्शन हो रहा है और यहाँ पे C कार्बन पे अटैक हो रहा है. एज अ रिजल्ट ऑफ this pi bonds are shifted this side. ये इंटरमीडिएट आप रिएक्शन देख रहे हैं. यहाँ पे N पे पॉजिटिव चार्ज आ गया है. सो और F बॉन्ड जो है यहाँ पे F जो जुड़ा हुआ है वो रिलीज हो रहा है, और while this group is getting attached here. तो यहाँ पे भी पॉजिटिव चार्ज है, यहाँ पे भी पॉजिटिव चार्ज है। पहले इस डायरेक्शन में इलेक्ट्रॉन्स शिफ्ट हुए हैं, देन अगेन पाई बॉन्ड इज बीइंग मेड in this intermediate structure. Now, final structure is like this. ये डाइनाइट्रोबेंजीन है और वो NH ग्रुप से जुड़ गया है while F is out. Then this F again attacks hydrogen of this NH. This should be NH, and as a result of which this derivative is formed, and this is yellow in color, but because of the presence of this at the terminal and terminal end, this will be hydrolyzed, and all amino acids will be released, and these two will be colorless, while this will be colored, and this can be obtained. These all can be run on chromatography, and this will be yellow in color, and this can be this can be released and collected and followed for the presence of, for the identity of this terminal amino acid. This reaction is used for protein sequencing, and one by one we can release this, and again we will know this amino acid and this amino acid and so on. Other applications are the alteration of enzyme activities of some enzymes. So it strongly inhibits the reductase activity of mitochondrial cytochrome B C1 complex. Addition of DNFB modifies amino groups and basic nucleophilic groups in amino acid residues, resulting in a decrease of both electron transfer and proton translocation in BC complex, BC1 complex. Then catalytic properties of fructose 1,6-diphosphatase is also altered by adding reagent at neutral pH compared to that at high pH, and it was shown by scientists that 4 equivalents of reagent per mole of enzyme increases the catalytic activity of fructose 1,6-diphosphatase by 3-4 times. Dinitrophenylated enzyme shows a change in optimum pH from 9.4 to 7.5. Also, the addition of this reagent results in a significant increase in Michaelis constant for both substrate and metal ion activator. It is suggested that this reagent alters the allosteric site of the enzyme, thereby induces a change in protein conformation, resulting in a change in activity. Finally, this can also be used to distinguish reduced and oxidized forms of glutathione and cysteine in biological systems, especially in blood and cell lysates. So, this is all for Sanger's reagent. And if you like my lecture, please like it, share it, and subscribe it.